Welcome to Mr. Chalk's Revision Tips.
When answering exam questions, there are a few key things to remember, right? So just remember, exam technique is a key part in preparation for any exams, and it's something that you'll pick up the more you go and you do past papers. Right, so many questions in exams are repeats of what will be in old exams. So going and looking at lots of generic questions beforehand is a good idea. When going and answering and questions and reading them, there's a few things just take into account. Make sure you go read the question at least twice so you don't make any mistakes when you're reading them. As you're reading them, go highlight key information. Don't make assumptions about what the question is asking. Make sure after you've answered it, you go check your work for daft errors that you might make. And if it involves going and answering something where there's some calculation, make sure that you write your workings out down. Right, let's go and have a look at the question.
In this video, we're going to look at transcription and how you answer exam questions on transcription in A-level biology exam questions. So, some key things to go and note just before we go and look at the question. So remember that most mechanisms of gene control or expression are done so by controlling transcription. Right, so remember, transcription is the synthesis or the production of messenger RNA. So that can involve things like chromatin remodeling. Remember, chromatin is basically the DNA. We can go and we can change the chromatin architecture that can go and cause various things to happen to it. For instance, it can go and prevent it being transcribed by stopping the RNA polymerase getting to it, or it can go and increase the rate of transcription happening by causing RNA polymerase to go and bind more strongly to it. So some of the key factors and things that we need to consider. We can't have transcription factors that go and bind to it, right? So these are substances such as proteins that go and contribute or go and bind to specific biochemical regions within the DNA. Right, so we can see just here we have a transcription factor. You can see that transcription factor there is binding to that specific point on the DNA, right? Because it's binding to that specific point on the promoter region, and we'll look at promoter regions in a minute, that will go and help promote the transcription of the genes further down the DNA. Right, we've got the promoter. So the promoter is our region of DNA that initiates, right, so starts to cause the particular transcription of a particular gene, right? And it's because you've got that initiation at that point, that's why the transcription factors usually bind onto them. We've got DNA regulatory sequences, right? So these control gene expression by forming DNA protein complexes within specific binding points. So we can see just here we've got that particular regulatory sequence, and we can see that we've got these gene regulatory proteins that are bound to it. If they're bound to it there, that is going to go and prevent the RNA polymerase from moving down the gene. We can have gene regulatory proteins, right? So these kind of recognize short stretches of the DNA double helix, and therefore can go and help transcribe a number of different genes in different regions, right? So you've got your regulatory sequence there, right? But lots and lots of genes will have a regulatory sequence that sits before the promoter sequence. Right, and we can have promoters quite importantly. Right, so promoters, sometimes we call them enhancers, are regulatory elements that do not code for proteins, most importantly. Right, but they can go and regulate the production or how much a gene is transcribed further down the DNA at a particular time. And finally, the last thing to consider whenever we're going and we're talking about our genes being transcribed, we need to talk about RNA polymerase. Right, so remember, RNA polymerase is the enzyme that is responsible for copying the DNA and going and producing that RNA sequence, right, during the process of transcription. So moving on then, if we want to go and look at this question, right, in this question it talks about, so gene transcription is controlled by a number of different factors, right, or a number of different transcription factors. So we can already see that it's talked about transcription factors in the question, right? So figure 16.1 shows how a transcription factor can control transcription, right? So everything about this, we need to go and look quite carefully at this diagram. So we start off, so we've got a hormone that's going to go and pass over this plasma membrane. Right, remember, hormones are lipid soluble in this case because it's passing straight through and it doesn't need some sort of receptor protein to bind to it. So that is kind of the first thing. Right, the second thing is that down here we've got an inactive transcription factor. So that's a transcription factor that hasn't been activated yet. Right? So when the hormone binds to it, it will go and activate it. Remember, when a hormone is binding to it, it will usually, or can quite often, bind to some sort of allosteric site on that transcription factor, which causes conformational change within it that goes and causes it to be activated. And then finally, we can see that our transcription factor will go and bind to a specific region on the DNA that the RNA polymerase is going to go and bind to. Right, so remember that if the RNA polymerase is binding to that, that's probably going to be some sort of promoter region, because that's going to go and promote the transcription of this gene further down the DNA. So if we go back to the question, if we're looking at naming site A, right, quite simply, site A is going to be a promoter or a promoter region. Right, moving further down, so using the information from figure 16.1, so what we've just talked about, describe how transcription can be controlled by eukaryotes. Right, importantly there, remember that prokaryotes are like organisms or cells with a nucleus. So some key things to talk about, like we've just talked about. So we're talking about the hormone, right, going and binding to that transcription factor. Right, you've got to be really, really specific in what it's doing. That is going to go and cause that transcription factor to be activated. I personally would go and talk about the hormone binding to an allosteric site on it and causing some sort of conformational change, which activates it. Then we can talk about that transcription factor binding to site A, or I will talk about the promoter region, which finally goes and causes RNA polymerase, so down here, so RNA polymerase to go and bind to that promoter region, and the RNA polymerase will go and transcribe that gene. So hopefully you have found that useful. If you've got any questions, then drop it in the comments below, and we'll be uploading a lot more. Right, good luck, folks.